Thank you so much, Sonia, for the introduction. And thank you for inviting me to Reef Talks. It's a pleasure for me to present part of my PhD thesis that focus on reef complexity and the application of underwater photogrammetry. And in this study, particularly, I use underwater photogrammetry to map reefscapes and to calculate new physical and ecological habitat descriptors. And then I explore the links between these habitat descriptors and specific fish groups with linear mixed models. So let's begin with the context of the study. I think most people in this virtual room know that coral reefs are one of the most diverse and productive ecosystems worldwide, despite their importance. If the actual management are not improved, coral reef is expected to be in very high or critical risk in a near future. One of the main characteristics of a natural ecosystem is the structural complexity, which can be defined as their three-dimensional structure. Also known as habitat complexity, this composite aspect plays a key role in the dynamics of the ecosystem, supporting their stability and the resilience. So tropical coral reefs are one of the most complex three-dimensional bioconstruction on Earth. Reef-building corals are the main engineers, organisms, and they largely shape the structural complexity of this ecosystem, providing shelter capacity, habitat, promoting the biodiversity, and supporting the stability and resilience of reef. In addition, reef structural complexity offers coastal protection that has been shown as more determinant feature than sea level rise to ensure the coastal protection in the near future. Other investigations show the conception of innovative reef restoration solutions to mitigate coastal erosion and flooding. For all these reasons, reef studies increasingly recommend the evaluation of the structural complexity to assess the ecological state of coral reef. So, methods to quantify habitat complexity of coral reefs began describing reef profiles and coral growth forms of different reef zones. Later, chain method was described measuring the reef rugosity, and this measure was later adopted to quantify structural complexity of coral reef, and then to explore the relationships between the rugosity and fish diversity. Fractal dimension theory was later used to describe coral reef topography and investigate the complexity of reef structures and at different spatial scales. Other methods combining different substrata characteristics were described also to quantify structural complexity. These multifactorial approaches allow to better identify relationships between habitat and reef biodiversity. So, since 2000, novel methods were adopted to describe the structural complexity of reef. Among them, airborne lidar, acoustic technologies, and hyperspectral and multispectral are new technologies that allow mapping and can describe a structural complexity of reef environment. Focusing on the fine scale, stereo-derived 3D models, and notably the application of underwater photogrammetry by structure from motion have become a powerful tool for quantitative benthic reef studies. So photogrammetry is the science of obtaining information about the properties of an object or an environment by recording, measuring, and interpreting photographic images. Structure from motion is a technique which allows three-dimensional reconstruction of objects from overlapping images. So principal steps of photogrammetry workflow are the image acquisition, the camera calibration that allow to obtain an sparse cloud, and the point cloud densification that from this point cloud densification, we can obtain different outputs. We can obtain a mesh, it's a 3D model, the digital elevation models, that are a grid of the elevation of a continuous surface, and orthomosaics that are a single mosaic image, product of several image collections and can be used as a map. These outputs provide the possibility to perform a large panel of measurements and mapping analysis. So underwater photogrammetry application began for industrial procedures. Archaeology researchers were also pioneers in adopting this technique to map their in their studies areas. In the marine biology field, the technique was progressively adopted to conduct physical and ecological analysis as morphometric measurements of organisms and to coral reef mapping. Other several studies were conducted over the last decade, proving the relevance of underwater photogrammetry for studying and surveying coral reef. So the improvement of management efficiency is the greatest challenge of the 21st century reef conservation. Novel reef survey technology can help to solve critical issues facing coral reef ecosystem, but several barriers must be overcome to maximize their impact. Among these technologies, underwater photogrammetry is particularly relevant to conduct quantitative monitoring for multiple spatial and temporal scales. So more or less, at the same time, the last 2 decades, life history traits of corals and reef fishes have been increasingly used to better understanding their ecosystem roles. Also, fish assemblages have been increasingly studied from a functional point of view through functional entities which are defined by shared life history trait. To better understanding the complex relationship between habitat and fish assemblages, it's necessary to explore the links between new habitat descriptor and fish groups, in particular, thus ensuring key function of the ecosystem. So underwater photogrammetry provides new tools to define new quantitative habitat descriptor. For instance, here, a study that show new physical descriptors and explore the correlation with fish diversity or abundance. So in this context, the study objective was to identify the complementarity and redundancy among new quantitative habitat descriptors obtained by photogrammetry, and to explore the relationships between this descriptor and the diversity, abundance, and biomass of fish groups, ensuring key function of reef ecosystems. So our study was conducted in 24 outer reef slopes sites around 3 islands, Europa, Reunion, and New Caledonia. The sites were 3D model and the habitat descriptor were calculated from digital elevation models and orthomosaics. Here on the right, the quantitative habitat descriptor for 3 reefscapes, the most complex in the top, Europa, one moderately complex in New Caledonia, and the less complex reef in Reunion. The surface complexity here used at the reefscape scale, the slope, the fractal dimension, the total coral cover, shelter capacity were calculated from predicted code described in other study from a PhD thesis. After that, we calculate also the Shannon shelter index, the abundance of colonies, and the mean neighbor distance. Concerning fish assemblages, they were evaluated by a remote underwater stereo video and at Europa and New Caledonia. Underwater visual sensors was used in Reunion Island. The diversity, abundance, and biomass of fish groups were then calculated. The life history traces were compiled for each species, and we consider 5 of them: diet, size class, schooling, home range, and level in the water column. After that, we do the functional entities. That is a combination of 2 life history traits. For example, diet size, size schooling, level in the water column, and home range. So we use these 5 key ecosystem processes as a group of fish to explore the possible relationship with habitat descriptors. So here, one of the main results of this study, the Spearman correlation rank, used to explore the complementarity and redundancy of habitat descriptors. The correlation graph confronting the habitat descriptor here in the top. In the left of the graph, the habitat descriptor from the digital elevation model. So the slope, the fractal dimension, and the surface complexity. And at the right of the graph, the descriptor used from the orthomosaics. For this graph, we keep only 7 habitat descriptors. We selected by a coefficient of Spearman coefficients and measuring the level of correlation. And we use also the variance inflation factor that measuring the level of multicollinearity. So in dashed line, we have the 7 habitat descriptors that we use in linear mixed models. So as I say, we use a threshold for each parameter. And here the linear mixed effect models performance for predicting the diversity, abundance, and biomass of 4 key functional entities: preys, planktivores, grazers, and predators. So here what I want to show is that models were used new habitat descriptors, and particularly 3D habitat descriptors were more performant, and notably for predators. Biomass of predators was the more performant model that use shelter. That use total shelter capacity, and the model was improved. So it's the same for for the preys in the abundance and biomass. And planktivores was also a model that was improved by the surface complexity. So to sum up, we explore 4 groups of habitat descriptor, and we identify the most complementary or less redundant. The idea was to identify the relevant and time-efficient descriptor for monitoring reef habitats. And then looking the linear mixed models, we confirmed that coral cover is a historical descriptor that is used to explore or to predict fish assemblages, and is an important predictor of the structure of fish assemblages, and did not contribute to the prediction of plantivore and predator metrics in our study. Adding surface complexity and 2 novel 3D descriptors, shelter and channel shelter index in the models. Sorry. When, s'il te plaît, désolé, je suis en ligne sur un séminaire. Désolé. Sorry for that. Adding surface complexity and 2 novel 3D descriptors, shelter and channel index in the model allowed us to explain the metrics of 4 key ecosystem functions ensured by fishes. So our results show the added value of coupling fine-scale 2D and 3D descriptors to predict the metrics of key fish functions. So the limits and perspective of the study. The principal limit is that we have two different fish surveys that was also good for our analysis. So it's better to have one type of surveys. And for the perspective is to continue to explore these new key habitat descriptors and to try to apply these new habitat descriptors for the monitoring programs in the future. So I would like to thank all my collaborators, founders, and partners that make this work possible. And thank you so much for your attention.